Hello, and thank you for joining me today. My name is Courtney Anderson, and I am a senior scientist in R&D here at ACD. Today, I will discuss the visualization of LGR5 positive stem cells and the immune response in the inflamed mouse colon using the RNAscope assay. The intestinal epithelium is a multitasking tissue that must achieve efficient digestion and absorption while maintaining an effective barrier against agents in the lumen. The harsh luminal environment of the intestine induces a high rate of cell death, which requires the daily self-renewal of the intestinal epithelium throughout life. This self-renewal is driven by a small population of adult stem cells that reside within specialized niches. The organization of the intestinal tissue is highly adapted to both its function and maintaining its integrity. Both the small and the large intestines have similar cell populations, but slightly different structures. Because the small intestine is primarily responsible for absorption and digestion of food, it has villi that protrude outward toward the lumen and contain the absorptive cells. In addition, both the small and the large intestine have crypts that protrude inward. The base of these crypts contains cells known as the crypt-based columnar cells, or CBCs, and this is where the intestinal stem cell population resides. We first wanted to visualize the multiple cell populations of the intestine. We used several RNAscope probes designed to detect known markers of each cell population in the small intestine. Enterocytes were detected with intestinal alkaline phosphatase. Goblet cells were detected with mucin 2. Enteroendocrine cells were detected with chromogranin A. Paneth cells were detected with lysozyme 1. And finally, the columnar-based crypt cells, where the intestinal stem cell population resides, were visualized with LGR5 and olfactomedin 4, or OLFM4. Okay. And this is just those same cell populations identified by their respective markers using the RNAscope assay at a higher magnification. Simultaneous visualization of multiple intestinal cell populations is possible using the RNAscope multiplex fluorescent assay. Here we can detect enterocytes, enteroendocrine cells, and the crypt-based columnar cells in the same small intestine section using probes for the markers intestinal alkaline phosphatase 1 in green, chromogranin A in red, and LGR5 in white. We next visualize the intestinal stem cell population that resides in the base of the crypt using the RNAscope multiplex fluorescent assay. Shown here are known markers of the intestinal stem cells with LGR5 in white, ASCL2 in green, and OLFM4 in red. The three signals co-localize in cells that are located primarily at the base of the intestinal crypt. The LGR5 positive intestinal stem cell population allows the epithelium to adapt to regenerative cues such as nutritional state and chronic inflammation, including intestinal bowel disease, also known as IBD. IBD includes diseases such as Crohn's disease and ulcerative colitis. Intestinal organoids generated from adult ISCs are a promising treatment for IBD, yet the interplay between inflammatory immune cells and ISCs remains to be elucidated. Therefore, we sought to visualize the intestinal stem cell population in the inflamed colon. To achieve this, we induced intestinal inflammation or colitis in mice using the chemical TNBS for 1 to 6 weeks. We also included 2 control groups, one that received no injection and another that received vehicle injection containing 50% ethanol. Indications of colitis included a reduction in body weight, changes in stool consistency, and blood in the feces and at the anus. Shown at the bottom are representative images depicting normal colon from control mice and inflamed colons from TNBS-induced mice, which contain large regions of inflammation. Colons from all mice were formalin fixed, paraffin embedded, and then sectioned. The RNAscope assay was performed to examine several stem cell and inflammatory-related markers. In this presentation, I will show a subset of these markers, but you can find the complete data set on our website. To ensure good quality RNA in our samples and no background due to poor fixation, we tested each sample with the negative control probe DAP-B and the positive control probe PBIB. All samples showed no background with DAP-B, while robust signal was detected with PBIB, indicating high quality of our samples. We first examined 3 markers of the intestinal stem cell population in the mouse colons, including LGR5, ASCL2, and BMI1. As you can see, these 3 markers are expressed at various levels in the crypt, with LGR5 and ASCL2 primarily expressed at the base of the crypt, while BMI1 is expressed throughout the entire crypt. However, in the inflamed region, LGR5 was strongly detected, while little to no expression of ASCL2 and BMI1 was detected. Stem cells are known to be controlled by the local microenvironment, or niche. Wnt signals emanating from the niche act as self-renewal factors for stem cells in many tissues, including the intestine. Therefore, we next examine the expression of several genes in the Wnt beta-catenin pathway. Shown here are axin 2, beta-catenin, and TCF4. In the control and non-inflamed colons, axin 2 expression was primarily detected at the base of the crypt, but also along the entire length of the crypt, as well as in the inflamed region. Beta-catenin was robustly detected throughout the entire crypt, as well as in the inflamed region. TCF4 was also detected throughout the entire crypt, as well as in the inflamed region, albeit at a lower level. We also examined the localization of receptors and ligands in the Wnt signaling pathway. In this image, we showed the visualization of the ligand Wnt3A and the receptor Frizzled7 in the control and non-inflamed colons, as well as in the inflamed region. And in this image, we showed the visualization of the ligand R-spondin3 and the receptor LGR5 in the control and non-inflamed colons, as well as in the inflamed region. This is a higher magnification image of R-spondin3 and LGR5, and interestingly, what we noticed is expression of R-spondin3 is primarily in the submucosa and muscularis mucosa, while LGR5 is primarily in the neighboring mucosa at the base of the crypt. IBD is related to an excessive uncontrolled immune response, and defects in regulatory T cells, known as Tregs, have been implicated in the pathogenesis of IBD. Therefore, we wanted to visualize this population in the inflamed colon using the RNAscope assay. Shown on this slide is a panel of genes marking the Treg cell population, including FOXP3, IL-10, and CD4. For all 3 genes, little to no expression was detected in the crypts of control and non-inflamed colons. However, robust signal was detected in the inflamed region for all 3 genes. In order to confirm the presence of the Treg population in the inflamed region, we performed the RNAscope multiplex fluorescent assay to simultaneously visualize these 3 markers, with IL-10 in green, FOXP3 in red, and CD4 in white. As shown here, we can see co-localization of these 3 markers in the inflamed region, suggesting the presence of regulatory T cells in the inflamed colon. We further characterized the cell population by performing the RNAscope ISH assay for IL-10 and FOXP3, along with IHC for CD4 on the same slide, referred to as dual ISH IHC. As shown here, we can see co-localization of IL-10 in IL-10 in green and FOXP3 in red, mRNA, with CD4 protein in white in the inflamed region. Lastly, we examined the expression pattern of the cytokine TNF-alpha and its receptor TNF-RSF1b in both the non-inflamed and inflamed colon. As seen here, we detect very low expression of both the receptor and ligand in the non-inflamed colon. However, there is very robust expression of both genes in the inflamed region. So in summary, today I've shown you that the RNAscope assay can be used to identify specific intestinal cell populations, visualize intestinal stem cells with morphological context in the intestinal crypt, and assess the relationship of intestinal stem cells to inflammatory cells. The RNAscope technology can aid in elucidating direct effects of inflammatory cues on the intestinal stem cells and their niche during pathogenesis of IBD and other inflammatory diseases. It can also aid in developing potential IBD therapeutics, such as tracking stem cell therapies currently being pursued for IBD. For more information on this study and the RNAscope technology, please visit us on our website at the addresses shown here. Thank you very much.